A timely question for clinicians and basic researchers regarding Huntington's disease is to understand the relationship that may exist between altered sleep and cognitive disorders that are associated with the disease. In order to understand how sleep is potentially modified in Huntington's disease, we have implanted a mouse model of Huntington's disease called R61, transgenic animals, with electrodes to record the electrophysiological activity. And we have made several observations. The first one is the continuous presence, the generalized presence of epileptic activity, electrophysiological activity, which in all animals recorded, even in those in which we could not see seizures behaviorally. And in fact, this may explain why this epileptic disorders has been probably underestimated up to now in Huntington's disease, at least in mouse models. And actually, the same thing happened with Alzheimer's disease when a few years ago it was identified also some epileptic component from electrophysiological recordings, while they had not been observed behaviorally. And so, therefore, there is this epileptic component inside Huntington's disease models, at least in the mice, which can by itself explain some perturbations of neural networks. Our second observation relates to the segregation of brain states. Normally, brain activity is very different between awake and sleep, or at least between active states as opposed to slow wave states of brain activity. And in R61 mice, what we observed is the intrusion of active patterns of activity, of cortical activity, within the slow wave state, which is absolutely abnormal. Because these cortical activations happened at moments when they should not, we call them ectopic. And in fact, they last for a few seconds, and they recur several times during the slow wave state. From electrophysiological point of view, these active states look quite normal, except that the hippocampal network seems to be hyperactive because it concentrates, because these events concentrate epileptic intellectual activity, as well as some bursts of activity, which we call ripples, which are supposed to be very important for the consolidation of information in the hippocampal network during slow wave state. Slow wave sleep is therefore altered by temporally misplaced cortical activation in R61 mice. With a group of clinicians, we are now going to investigate whether the same occurs with patients. Our third observation is that these ectopic activations are blocked by atropine, a cholinergic antagonist. And what is interesting is that the ascending cholinergic system, which is essential in the cortical activations, depends under the inhibitory control of basal ganglia, which are known to degenerate in Huntington's disease. And so we may make the interesting hypothesis that dysfunction of basal ganglia in Huntington's disease, which is probably responsible for the motor disorders of the disease, would also interfere with the consolidation of memory and with the cognitive treatment of information during slow wave sleep. This hypothesis would allow to unify our understanding of the motor and cognitive components of Huntington's disease. And this is why we are going to test this directly, experimentally.